Hello everyone. Now today we're going to do a past paper review, which is a Bgcse Biology Paper 1, 2020. Now before you start this examination, one of the very first thing that you want to ensure that you do is to write your school number, your candidate number, and also your surname with your initials. The second most important thing to do is to ensure that you read through the instructions carefully and also follow the directives of the person who is conducting the examination. Now let's jump into our first question. And our first question reads that which characteristics are common to all living organisms? Now always read through the options carefully and you can eliminate the incorrect versions. And so let's look at option A. And option A states that breathing, excretion, growth, and irritability. Now the thing is, not all organisms are capable of breathing. Some organisms will take in gases and also release gases by diffusion or simple gaseous exchange. For option B, we have excretion, growth, irritability, reflex action. Not all organisms will be able to carry out reflex action because not all organisms contain a nervous system. For option C, we have growth, irritability, photosynthesis, and reproduction. Not all organism is capable of photosynthesizing because not all organisms contain chloroplast. Then option D is our only correct answer. And remember that all organism is capable of responding to stimuli, which is irritability, able to excrete, which is removal of metabolic waste, and all organisms are capable of growing, and all organisms are also capable of reproducing. Let's now go to our next question. And for this question, it reads that the scientific name for the sea grape tree is the Coccoloba uvifera. And it's asked now, what do the two words in the name Coccoloba uvifera represent? Now, all scientific name, a matter of fact, includes two names, which is the genus and the species. So without any question, without any doubt, we know our answer here will be option A. What is also important for you to remember is that the genus is written starting with an uppercase letter and the species starts with a lowercase letter. That's very important for you to remember, okay? So just in case you see the scientific name again, you know what to do. All right, let's now go to our next question, which is question number 3. And question number 3 reads, which of the following includes organisms of only one species? And so if you look at community, communities cannot be the correct answer because community is a collection of different population. All right, ecosystem is even wider because ecosystem involve the entire community, in fact, different population interacting with even the non-living aspect of the environment. Kingdom is a large group containing many different types of organisms in terms of classification. So the very specific group here is population. And so you can see a population of dogs, a population of human beings, and so on. Now for question number 4. It said, which is the correct definition for omnivores? And remember, an omnivore is an organism that eats both plants and animals. So option A is incorrect because it eats only animals. This is representing a carnivore. Organism eating both plants and animals will be our option B. And option C, an organism that eats only plants, and this one will be herbivores. All right, and organism that eats dead animals or dead organisms or any dead plants, really, those are decomposers, right, or detritivores. All right, so let's now go to our next question here, question number 5. And question number 5 states, in which ecosystem would a sea fan be found? And now I wanted to note that a sea fan is a type of coral. So definitely without any doubt, we know this must be the coral reef. All right, let's now jump on to question number 6. Question number 6 states that eagles have hook beaks that help them to ingest food. Now there's a very important word here, the word help. Okay? It said, what is this an example of? And so the hook beaks are helping the eagle to ingest. Now, I know the word ingest will show off a lot of persons here, because once you see the word ingest, you think about digestion. But they're asking you about having a hook beak for helping with the ingestion of the food. So this is a suitable adaptation. All right, this is a very good adaptation of the eagle so they can ingest food easier. All right, so be careful with the words, or please try to read and understand what the question is actually asking you before you start answering. All right, so let's look at number 7 here. So number 7 asks, what is the correct order in which organisms occur in a food chain? Now, without any doubt, you'll know that the food chain must start with a producer. So option A and B, they are out already because they're not starting with the producer. The next organism after the producer is the herbivore. And so we know by this that this will be our option D. So you have the producer, the herbivore, and then carnivores. All right, question number 8. It said, which is not an important marine resource in the Bahamas? And so if you look at conch, everybody know that conch is famous in the Bahamas. Lobster indeed. Nassau grouper, absolutely. So the salmon is the only one that is not an important resource within the Bahamas itself. Now let's now go to question number 9. And question number 9 reads that which pollutant can cause eutrophication if dumped in an open body of water? Now, what is eutrophication? Now, eutrophication is the excess growth of algae when nutrients are dissolved in water bodies. And so we're going to look at the options that can actually deposit nutrients within a body of water. And looking through the options, you realize that aluminum cannot be the answer. Cardboard boxes cannot be. And of course, the option here has to be fertilizers, because fertilizers will add nutrients to water bodies. And what that will cause is causing algae to grow really fast and rapid. And that process is called algal blooming. So eutrophication is when the excess growth of algae in the water because of dissolved nutrients. All right, so that will be our only option right there for question number 9. Now let's go to question number 10. All right, let's go a little zoom out in that so we can read it properly. All right, so question number 10 states that limiting the amount of organisms that could caught at a specific time of the year is referred to as which of the following? And if you're limiting any catch of any organism, what you're trying to do is to preserve that organism. So it is a method of conservation. So definitely option A will be our answer in this case. All right, so conservation is indeed. All right. Diversification is definitely when you have a lot of organisms, different types of species, matter of fact, in an ecosystem. Endangerment is closer to extinction. And eutrophication, we just mentioned that excess growth of algae in water bodies due to dissolved nutrients. All right, question number 11. This one now said, which nutrient is broken down by the enzyme pepsin and trypsin? Now, I want to look at this question now carefully and remember that pepsin and trypsin, they are example of proteases. In fact, pepsin is found within the stomach and trypsin is produced by the pancreas. And so carbohydrates, if you think about it, carbohydrates are broken down by carbohydrases, is a general name of those enzymes. And a typical example of this, or a general grouping of this, is the amylase. Okay, so amylase will break down carbohydrates, and we have different types of carbohydrates. We have like maltase, right? We have like sucrase, right? But generally we say amylase. And fats are broken down by lipase. So lipase are responsible to break down fats. Proteases, example pepsin, trypsin, break down proteins, as we talk about that just now. And vitamins are generally absorbed directly in the body. You don't need to break down vitamins anymore. So they are absorbed directly into the bloodstream. All right, so now we'll jump on to our next. Okay, so let's highlight this real quick before I forget. So the answer here is proteins. All right. All right, so now let's jump into our next question here, which is question number 12. All right, so let's go to question number 12. All right, so this one here is saying that the question number 12, 13, and 14 refer to the diagram of the cell. And so what we want to do now is let's quickly label this so at least we know what we're doing. And because it's going to ask about these label structures. And so let's look at the labelings real quick. And so this structure right here, this will be our nucleus. Okay. This structure right here, which is R, that will be our cell wall, all right, which is the outermost layer of a plant cell. And structure S will be our plasma membrane or our cell membrane. Okay. All right, and structure P, that large, almost centrally located structure, that will be our vacuole. And then we have our structure T. And if you notice, there are a lot of these structures within the plant cell. So we definitely know that this represents our chloroplast. All right, so now we have all our structures. Now what we can do now is to go to the question and try to answer them as best as possible. All right, so now let's go to question number 12, because number 12 is asking us which label on the diagram shows the part of the cell where substances enter and leave the cell. Whether this is a plant cell or animal cell, the same structure does the same thing. And this is our cell membrane. So our cell membrane here is option C, which is structure S. So option C will be your answer. Again, the cell membrane or the plasma membrane controls what is entering and what is leaving any cell. All right, now let's go into our next question. Question number 13, still refer to the diagram. So we're going back to the diagram to see what this is. And so it said which organelle transfers energy from the sun into energy in sugar. So we're looking at what structure is converting the solar energy into the energy within the sugar. So it's a chemical energy. So as we know, we're looking for where photosynthesis is taking place. So let's go back up there and see which structure that is responsible for that photosynthesis. And the structure that is responsible for photosynthesis is the chloroplast. And so question number 13 is definitely D. So structure T, let me just highlight that real quick. All right, so, yep. All right, so let's go back and check. So option T is our chloroplast. Absolutely. Let's go to number 14. And number 14 is also referred to the diagram as well. And I said, what would happen to structure P if the cell was placed in a concentrated salt solution? It doesn't matter if it's salt or sugar, really. If the solution is concentrated, what will happen to structure P? What is structure P? Let's go back to it and you'll see exactly what structure P is. So structure P is the vacuole. Remember that the vacuole stores water, all right, plant food, and also waste. So if the cell is placed in a concentrated solution, and remember that a concentrated solution is called a hypertonic solution. So what will happen to the cell or the vacuole in this case is that the vacuole will lose water because the content of the vacuole will be more dilute compared to the solution outside of the cell. So in this case, the cell will shrink and shrivel because of loss of water by osmosis. Option A and D are pretty much the same thing, because if you notice it, they are turgid or swell and burst. That means they're hypotonic solution. And option B will be isotonic solution, meaning the cell remain the same. What goes in comes out in terms of the movement of water. Let's look at question number 15. And so number 15 states that a pregnant woman must select a meal for lunch. Which selection best meets her nutritional needs? And remember, she is pregnant, so she is obtaining nutrients to maintain herself and also the developing fetus. And so if you look now carefully at option A, it states that chicken sauce, which is good because it's protein and of course other things that are cooked within the sauce. And Johnny cake is a good source of carbohydrates, and definitely she needs some form of energy. But coffee is not such a good choice because coffee will produce or supply her with caffeine. And caffeine is not the best option for a pregnant female. Yes, she'll be up for too long, her body will be undergoing the stress, and the caffeine as a drug could affect the developing fetus as well. Option B, fried chicken, macaroni, and water. In terms of fat, all right, in terms of fat, then because of the fried chicken, you may not want that to be a good choice. And if you look at it again, you only have carbohydrates and protein and water, which is not a totally bad choice, but probably too much fat maybe in this. So let's hold on to B a little bit to see if there's a better option. Now, option C, meatloaf. And meatloaf, there's carbohydrate, which is the loaf part of it, and inside of it is meat, which is protein. So you have carbohydrates and protein here. Garden salad, which is nice, absolutely, because you get your vitamins and your minerals. You also have milk, which milk produces the calcium, which is the fetus is developing, making bones and all of that. And also a small amount of protein. So this one looks better than B. So let's hold on to this one. Let's look at D. Turkey sandwich. That's absolutely a good one because the meat from a turkey, the bread, which is carbohydrate, the chip, not such a good choice, even though the chip will produce carbohydrate. And of course, soda will make this one really a terrible choice. So we know that option A and D are pretty much out already. All right. And so our best choice between B and C, it has to be C. Okay. Because of the many different nutrients that they get, the carbohydrates, the protein, the vitamins, the minerals, the calcium. All right. And also other dissolved nutrients. So this is absolutely the best choice there for number 15, which is option C. Now let's go to question number 16. And in question number 16, it reads that what term collectively describe the following substances: thickeners, colorings, flavorings, and dyes. And by looking at them, you know, these are things that they add to food to make them whether taste better, look better, and even for a longer tasting and appearance generally. All right, so the fact that even use word additives, right? They add these to food substances, right, to make them look better and so on, and even taste better. And so number 16 here will definitely be A. They're additives. Okay, they're additives. Let's go to number 17. All right, and number 17 here we have that microbes. So we look at this now carefully. It said microbes are used in the production of various items. And when we say microbes, we talk about microorganisms, whether it be bacteria or fungi. All right? Protists or whatsoever. So we're talking about microbes, small organisms. Now, the thing is this now, it said what percentage of the items listed above use yeast in their production? All right, so for cheese, not quite. Beer, yes. Production of alcohol, because yeast will undergo alcoholic fermentation. So beer and wine pretty much on the same category. Bread is used. The yeast is used in bread. And the function of yeast within bread making is really to respire and to produce carbon dioxide that will cause the bread itself to rise. All right, and that's why the bread becomes soft and fluffy, because of the production of carbon dioxide. Now, the question asks, now what percentage of the items listed above use yeast? And looking at percentage, so out of the 4, we already identified 3 out of the 4, which is all of these 3. So 3 out of 4, that is definitely 75%. So we can clearly state that for this option here, that is 25% of the products use yeast. Okay, so question number 17 is definitely C. Let's go to question number 18. And so question number 18, 19, and 20 refer to the diagram of the human digestive system. This one is labeled for you, which is quite nice. All right, let's kind of go into it a little bit so we can see more of the question at once. All right, so we have the mouth, we have the stomach, we have the pancreas, we have the large intestine and small intestine. So question number 18 now asks, which structure on the diagram is the main site for absorption? Notice the key word here, absorption. So we're looking for absorption. Now remember, and you're careful with these words, because I know sometimes students may kind of misunderstand what the question is actually asking you. And so you want to interpret this properly. Absorption versus reabsorption. And those are the 2 words that are generally misunderstood. And so absorption is always taking place inside of the small intestine, right? Because that's where digestion also ends, in the small intestine. Now, reabsorption of water will take place in the large intestine. So please not get those confused. So absorption takes place in the small intestine. In fact, the small intestine is where digestion will end. All right, so now let's look at number 19. So question number 19, all right, it states now which 2 structures labeled on the diagram produce amylase. Now, a question before this, when we talk about enzyme, we actually mentioned them, right? So now let's go back again, amylase. So amylase is used to break down carbohydrates. So there are 2 places that carbohydrates are broken down. And so if you think about it, we have carbohydrates. We don't need to go back to the diagram since they have the words here. So the large intestine, there is no digestion taking place there, right? So no, that one is out. All right. And notice the keyword is a produce, right? It's a produce. That's the keyword here. So the pancreas produces 3 types of enzymes, which is our trypsin. Okay. So trypsin is produced by the pancreas, amylase as well, and also lipase, right? In the mouth, we have salivary amylase. So we have pancreatic amylase and salivary amylase. So we know that option B will be our answer right here. The stomach is what produces pepsin. All right, so definitely that's not the answer again. So option C and D, they are out because of the presence of the stomach. Stomach does not produce amylase. It produces pepsin instead. All right, and of course, the large intestine is not included in the production of enzymes. All right, so by process of even eliminating these options, we know that option B is our correct answer. All right, so let's go to question number 20. And in question number 20, it states now, in which organ does chemical digestion of proteins begin? Notice the key word there, chemical digestion of proteins. Now, you may see question comes up to say where the digestion of protein begins. If it was just a blanket statement to say where the digestion of protein begins, then that's the mouth, because you do it by physical digestion or mechanical digestion. But the key thing here is that they stated chemical digestion of proteins, right? So if you're talking about chemical digestion of proteins, then you know our stomach will be the correct answer right there for question number 20. All right, so now we understand that. All right, so let's now go to our question number 21. All right, let's get a zoom inside of this one, zoom in a little bit so we can actually see what's going on here. So question number 21, so the diagram shows a stripe leaf from a plant. So notice you have a white stripe, you have a green stripe, right? And so let's go down to the statement here and then the question. And so here it said that the plant was kept in bright light for 6 hours. Let's pause there a bit. Bright light, if a plant is in bright light, it therefore means that the plant was what? Photosynthesizing. That's the key point right there, right? Now we said a leaf was taken from the plant and the chlorophyll removed. So once you remove chlorophyll, you know what you're going to do next. This is actually a part of your starch test experiment, right? So they said the leaf was then tested for starch using iodine. They said which answer best described the correct result of the test. Now remember that only where you find chlorophyll, you have photosynthesis taking place. And once you have photosynthesis taking place, then you have sugar producing and eventually starch. Right? Starch is a stored component. And so let's now give a little bit of reasoning onto this. All right, the white stripe, there's no chlorophyll, but the green stripe, chlorophyll is present, right? And so since chlorophyll is present in the green and not in the white, what do you expect to happen here? Remember that iodine is a yellowish brown substance and it turns into blue-black if starch is present. So let's look at option A. It said the green stripe turns blue-black. That sounds logical because the presence of the chloroplast. White stripe, well, it remains. It's I say it turns, but we understand what is happening here. So the white stripes, the color of the iodine remains yellow-brown, right? So that seems quite good. Option B doesn't check through them to make sure that we are answering it correctly. So here it said that green stripe turns blue-black, it is true. White stripe turns blue-black, it is not true. Green stripe turns yellow-brown. White stripe turns blue-black, not true. So definitely we can just stop there because we know definitely our answer here is option A. Okay. Now let's go into now question number 22. Our question number 22 now, so the diagram shows a cross section of a part of a leaf. And so if you notice the part of a leaf, you have the upper epidermis. Well, I must point this out right here because again, you have to be careful. Even though this is labeled the upper epidermis, you know that the upper epidermis is covered by a waxy cuticle, right? But they're just indicating the upper epidermis at this point. You have a lower epidermis as well. And notice the openings at the lower epidermis, those are the stomata. And of course, you have layer X. So be careful with this now. And then it's asked now, which type of cell is found in layer X? So what you want to think about, which type of cell can be found below the epidermis? That's the key thing that you need to think about. Which type of cell can be found below the upper epidermis? So let's look at the options to see exactly what the options are. All right, and say, remember now, so now we have diagrams to present these cells, not even names. But I want you to remember something. First, you have the waxy cuticle. Then the upper epidermis, then you have the palisade mesophyll layer or the palisade parenchyma. And so the palisade mesophyll layer is what contains the palisade cells or the palisade mesophyll cells. Now, looking at all these cells, we know for sure that the palisade mesophyll cells are the most elongated one. In fact, they are also the one that contain the most chloroplast. So these spot right here, they are the chloroplast within the palisade. So option A will have to be our choice. Okay. All right. Option D looks like a spongy mesophyll cell because it's kind of more circular and not as big or elongated as option A. Okay. For the mere fact that option A, they are elongated and contain a lot of chloroplast, that is our option. Okay, so now let's jump into our another question here, which is question number 23. All right, it said, what is the first product of photosynthesis in a leaf? All right, by this we know that this has to be glucose because glucose is the end product of photosynthesis. Remember that, okay? So 23 is B. All right, cellulose is what forms structures, especially those rigid supporting structures in the plant, and proteins are not formed by photosynthesis, right? Because proteins required nitrogen. So in the nitrogen cycle by bacteria and so on, nitrifying bacteria and all those are responsible to help with the nitrates needed by plants so they can convert the nitrates into proteins, right? Starch is stored. So starch is formed after glucose, and then cellulose is much more complex. So it's even one of the last that is formed within the plant itself, right? All right, so let's jump on to question number 24. All right, question 24 seems very interesting here. And 24 is asking us that a celery stalk is placed in a beaker of a blue dye. I hope you can see this clearly. It said once the dye reaches the leaves, the stalk is taken out and cut as shown in the diagram. So they take out the stalk and then they cut the stalk right here, okay? Now let's look at what they're saying in terms of the option here. It said which diagram shows the appearance of the cut end of the stalk. So notice it said we're looking here for the appearance of the what? The cut end of the stalk. Now if you cut the stalk, what should you see in it? Now remember, we have dyes, right? So since we have dye in the container, we have dye in the container, right? So what will happen to the dye? In fact, the question I really want to ask you, in what part of the stem or the stalk will the dye travel through? That's the question I really want to ask you, right? So think about it now carefully. The dye will travel in the liquid, which is the water. Now, water is transported through which vessel? And that is the xylem vessel. So water is transported through the xylem vessel, and the dye is dissolved in the water. Okay? So now the idea is now, where are the xylem vessels more likely located? In stems, the xylem are more in the inner portion, and the phloem, they will be on the outermost portion. So option A is totally out because it's not showing any transportation of the dye at all. Option B looks really good. Okay, option B looks really good because these spots will represent the dye that will be more likely in the xylem. Let's look at option C. Option C, the dye are on the outermost part, which indicating the phloem. The phloem is on the outermost part in the stem. So this dye will not travel through the phloem, it travels through the xylem because it dissolves in water. And of course, all over the stem is totally also incorrect. So definitely our answer here is what? B. And remember, the reason why option B is the correct option is simply because the dye will travel through the xylem vessel. Why? Because the dye is dissolved inside of the water. Okay? All right, let's go to question number 25. And we're halfway through the paper. And so question number 5 states that which two substances are transported in the phloem? Okay, now think about this now keenly. there's one substance that everybody should know, is that the phloem transports plant food, mainly, not only, but mainly sucrose. Okay, so let's look for the options with sucrose, which is the sugar. And so we are now stuck between option B and option D. So option A could not be the answer here because we're talking about fats, right? And remember, fats are also stored within the plants as well. So it's a storage molecule. Amino acids are obtained from proteins. So definitely amino acid can travel. So again, we are stuck with option B and D because of the presence of what? Sucrose. Now let's see which is the most appropriate one. Now, one thing you need to remember, that starch is a stored substance. So starch is not traveling through the plant. It's actually what? Stored. So our option here has to be option B, simply because amino acids are molecules that are able to transport throughout organisms. All right, because when proteins are broken down, or even when molecules come together in terms of anabolic reactions, they will end up forming proteins, right? In catabolic, like in animals, they will break down the proteins in forming amino acids. So very importantly, we need to understand that amino acids are able to travel through all living organisms. All right, so 25 is definitely B. And so now let's go to 26. Now, question number 26 now states, which statement about plant transpiration are correct? Looking at the transpiration of plants now, that's very key. All right, so notice the two columns. First and foremost, it says plants transpire most when, and plants transpire least when. So we're looking between the difference between most transpiration and the least transpiration. First thing, will plant transpire the most when it is dry? That's sounding good, because if here is dry, which means low humidity, then yes. Will plant transpire the least in high temperature? Absolutely not. So that is not a correct answer at all. So looking at now option B, plant transpire the most when light intensity is high. Yes, because the stomata will become open. Remember, light stimulates the opening of stomata, right? And least when the air is humid. Humid means a lot of water is in the atmosphere. So if a lot of water is already in the atmosphere, the plant can't lose a lot of water because the air is already saturated with water. Okay, so this is a good option here. Option B is good for this. Now, option C, light intensity is low. Definitely not correct. So there's no even need to go any further to talk about low light. Low temperature is good for least transpiration. Absolutely. For option D, temperature is cold, transpire the most? No, absolutely not. So for 26, our option here remains what? B. All right, and again, it's always good for you to check through the options as well, just in case you may miss something or you realize that there's a better option, because sometimes the answers can be really, really close. Do not just rush and answer one question and just, okay, move on. No. Look through the options carefully before you make your decision. All right, for question number 27, here it reads that the diagram shows an experiment to investigate respiration in yeast cells. Now, what you notice here, you have yeast in sugar solution, which is great. And this is the start of the experiment, and there's a balloon here. And now, after the experiment, so after 24 hours, you notice the balloon was inflated. And here you have yeast, sugar, and a new compound. So what they're asking you now is that which gas is produced and which new compound is present after 24 hours. As we talk about this in the bread making process, we already stated that once yeast respire, yeast will produce carbon dioxide, right? So the gas produces carbon dioxide because remember, yeast respire anaerobically. So we know that these options are incorrect. C and D, they're out, right? Also remember that yeast is used to produce alcohol, which is ethanol, which is drinking alcohol, right? And so without any doubt, we know that this answer here will have to be our option, all right? And so option A is correct. The gases produced is carbon dioxide. The new compound formed is ethanol, okay? And that's the fermentation of yeast using yeast, and that is producing alcohol, which is alcoholic fermentation. Now, let's look at question number 28. Now, number 28 states now, or asks, our aerobic respiration and anaerobic respiration similar? How are these respirations similar? Now, we should know the difference by now. And so when we talk about aerobic, we know that it requires some form of what? Oxygen. And we talk about anaerobic, it is without oxygen. Okay, let me just go in a little bit more so we can get a better view of the question. All right, so for this now, it's that both involves the breaking down of glucose. How that is sounding to you? Yes, all respiration requires what? Glucose as the source or the raw material. All right, so whether it be aerobic or anaerobic, glucose is needed. But let's look at B first. The both need a low concentration of oxygen. Absolutely not. Anaerobic is not requiring any oxygen, is without oxygen. C, both produce carbon dioxide in the muscle. Not true. Not true at all. Because what you also need to remember that there are two types of anaerobic respiration. You have the alcoholic fermentation, you have the lactic acid fermentation. Lactic acid fermentation does not produce any carbon dioxide, right? Just to point that out. And it said both produce alcohol in yeast. Not true. Okay, only anaerobic respiration will do this. So definitely 28 will be A as well. Okay, awesome. All right, now let's look at question 29 real quick. I need to zoom back into this so we have a better viewing of these options as well. Let's see how we could do it this way. Awesome, right. So that works. Now for question 29, it states that the diagram shows some of the structures in a human lung. It said, where is the carbon dioxide concentration highest? It says circle the correct letter. All right. And what I wanted to note right here is that this structure, matter of fact, you should also know this. This is the end of a bronchiole. And so you have the alveoli there. Okay. And so what is important here is that this is coming from a pulmonary artery. All right. Remember, the pulmonary artery is the only artery that carries what? Deoxygenated blood. The pulmonary vein is the only vein that carries oxygenated blood. And so what is also important, notice the arrow, right? Let me zoom into it a little bit so you see what I'm saying. The arrow is showing the direction of blood flow. So you know that the blood going to the lungs will be deoxygenated blood. What that means, a lot of carbon dioxide. What coming from the lungs will be oxygenated blood, which means rich in oxygen and low in carbon dioxide. So now let's look at this now carefully. The question asks, where is carbon dioxide concentration highest? Right? So remember, it's deoxygenated blood. So this have to be the one that is carrying blood into the lungs. So this has to be here. This is option A. Just to make a spin on this one real quick before we move on. B will be the most oxygen and least carbon dioxide. Let me erase that. I don't want to get confused with the option here, right? So again, in terms of the highest carbon dioxide, it is option A. The least carbon dioxide will be option B. All right, and the most oxygen will also be in this in terms of the blood itself, right? Because remember now, it's absorbing from the alveoli, going to the blood, then we transport it to the heart, and then it circulates around the body. Okay, but of course, here where you have the oxygen is being picked up, carbon dioxide being dumped, and so on. All right, so excellent. All right, so let's move on to our next question, and this is question number 30. All right, and so in question number 30, it's a diagram shows some structures in the human neck and thorax. so which means the chest cavity. And so we have Q there. I know they're going to ask something about Q based on how they label it. It said the line of tube Q has cilia. Remember, cilia are hair-like structures that will sweep dust particles out of the respiratory system. And cilia works with mucus. So the mucus will trap the dust particles or the foreign objects, and the cilia will help to sweep it back out from the system itself. All right, so let's look at what they're asking us here. And this is question number 30. It said, what is important? What is the important function of the cilia? And I guess I just stated that. So the important function here, as we look through it, helps to exchange help the exchange of gases, which is not the correct answer. Part B or option B is said to increase the internal surface area of Q, which is not true. To moisten the air, not true. And so here, option D, to remove dust and microorganisms, definitely by sweeping them out, okay? Now, 31, question 31. It said, which substance lost from the body by the kidneys, lungs, and skin? So the question is really asking you, which substance is being excreted by the kidney, lungs, and skin, right? So which is common between all three? Is carbon dioxide common between all three? Think about it. Carbon dioxide. No. Excess iron, because remember, ions could be in the form of salt too as well, right? So it's not true because the lungs is not doing that. Urea, not true again because the lungs will not get rid of urea. But water is indeed the correct answer because even when you're exhaling, you will release some water. If you try to blow or release exhale here in front of a mirror, you'd realize that the mirror will contain some form of moisture in it, right? Kidneys by urine, and water is in urine, and the skin is sweat. So water is common in all three. And remember, the water is a byproduct of respiration as well, so definitely. All right, so let's jump on to question number 32. Now, question number 32 states now, which is a function of the human blood system. So let's now probably going to get into it a little bit so you can see a little bit better. So which is a function of the human blood system? One, it states to transport eggs from the ovary to the uterus, not seeming so correct. B, to transport glucose from the gut to the liver, right? So this is a very important function of the blood because remember, at the small intestine is a part of the gut. The small intestine where absorption is taking place, glucose is being absorbed there, then it carried to the liver by the hepatic portal vein, again, which contains blood. So option B seems real good. Let's say option C, to transport partially digested food. Not true. Once you hear the word partially digested food, that cannot be a true answer. That is not found in blood at all. Imagine the condition of the blood to have undigested food there. Absolutely cannot be the answer, right? So option D, transport urine from the kidneys into the bladder. No. Remember what is happening there. That is taking place by a ureter, which is a tube. So option B is the correct answer for question number 32. All right, let's go into question number 33. And question number 33 here is saying that the diagrams show 4 different stages in one heartbeat. Okay, so 4 different stages in one heartbeat. All right, I think I need to go into that a little bit more. Okay, awesome. So you can see everything at once. Let me just create a mess here. Let me just erase that. All right, so we should be good to go now. All right, so yep. All right, so look at it now carefully. I want to show you all of them. All right, so we have P, Q, R, and S. Now, the arrow is showing the direction in which blood is moving. And so let's look careful what is happening here. You can notice how blood is actually flowing through the diagrams. Let's go into P and Q first, and I go into R and S. You can actually see, okay? So in P, the blood is going into the atria. and Q, they're leaving the ventricles, and R, they're moving from the atria into the ventricles, and then S moving out of the ventricles. Well, basically, if you notice here, it's going out and also coming in. All right, so let's now look at what is happening here, because S is the blood vessels, really. All right, so now look at the options or look at the question, really, because we didn't talk about the question yet. So the question states, what is the correct order for the stages after stage P? So stage P will be the first stage. So what happened after that? So if we go back to the diagram now, let's look at all of them. So after stage P, blood is going from the atria, and they must go into the ventricles next, right? So it has to be R next. If you think about it, R must be next. All right, and since going to R next, after you leave from the going to the ventricle, then it must leave from the ventricle, leaving out in the blood vessels, and then it'll be in the blood vessels afterwards, right? So let's see what we can pick up here from our RQ and then what? S, RQS. And that there is our option what? B. So option B is our correct answer. And remember again, it comes, blood will flow into the atria, then into the ventricles, then it will go out. All right, and then it'll be in the blood vessels, based on the blood vessel taking it to different parts of the body and so on. All right, let's look at our next question real quick. Question number 34. All right. In question number 34, all right, they said the diagram shows human blood as seen through a light microscope. It said if a person's blood is unable to clot, which component of blood is not functioning properly? Circle the correct letter. All right, so first you need to remember what is responsible for blood clotting. And that will be our platelets. All right, so if we should label these, structure A will be platelets. Structure B will be plasma. Structure C will be red blood cells. And structure D will be our white blood cells, especially phagocytes. All right, so question number 28 is 34, sorry, is definitely our A. All right, so now let's now go to our next question here. All right, and so what I want to do is to now look at our question number 35. And so our question number 35 reads that the diagram shows organs in which urea is produced. Let me get a little zoom on this one to get diagram a little better. So here now it's stated that the diagram shows organs in which urea is found, and so we have 3 organs here. We have P, Q, and R. And so the first thing is to identify what is P, and P is the liver, Q, which is the kidneys, and R is the urinary bladder. So the question asks which organ makes urea and which organ removes it from the blood. And so you need to remember that that urea is produced in the liver by a process called deamination. So what we're looking for here is that P will make the urea, right? While what is removing the urea here will have to be diagram or structure Q, which is the kidneys. Okay, so P making and Q removing. So our option here for question number 35 is A as well. All right, so now let's go to our next question. Our next question right here is question number 36. And for question number 36, let me just zoom it out of this. All right, so question number 36 is say that the diagram shows an experiment to investigate the response of a plant stem to gravity. And so what I want to focus on here, first and foremost, right? You notice the orientation of the plant cell. That's key. That's critical here. Now, it said that which diagram shows a suitable control for this experiment. When I say control, you want to make sure that all the factors remain the same and that you can use your control to compare any possible changes. So if you look at it carefully, look at option A, the plant is keep on rotating. So if you keep on rotating the plant and do it slowly, which is key right here, it therefore means that the plant, whatever the stimulus is, can be distributed equally around the plant, right? Option B is on one point. I notice the arrow going in, so it could be light going that side. C, turn sideways, light going this side, and D is in the dark. Now, the best plant right here that making the stimulus distribute all over the plant is actually option A. Okay, option D looks like a good choice as well, but the fact that if the light itself change, then it can affect different parts of the plant. But in option A, since the rotation is taking place, it therefore means that the stimulus will be distributed all over the plant, or you can say that the plant is exposed equally to the stimulus, and that's why A is a better choice. Okay. All right, so now we covered that. All right, let's jump into our next question. And this is question number 37. Now, for question number 37 here, it said now that a boy accidentally touches a very hot object and immediately takes his hand away. It said, in this reflex action, what is the effector? Remember, effector or effector cells, they are found within the muscles. Okay, they are found within the what? Muscles. And so the muscles within the arm will contain the effector cells because that's where the action will take place. And think about the word effector, that is what going to bring about the effect. So if you remember the order in terms of the cells that are involved in a response, you have the receptor cells in the sense organ, then you have a sensory neuron. After the sensory neuron, you have the relay neuron, otherwise called the interneuron. After the interneuron, you will go into the motor neuron, and from the motor neuron, you go into the effector cells. And effector cells are found within the muscles that will bring about that action. Okay, so question number 37 is definitely C. So let me quickly highlight that one. All right. Now let's jump into our next question here, which is question number 38. Now on question number 38, now it said that the diagram shows a section through the human eye. It said which label part prevents the internal reflection of light in the eye. And so the different parts that are shown right here, this here is the aqueous humor. So the aqueous humor, all right, we have the lens, that is A. So A is the lens. All right. Let me see if I could move this where so you could see it a little bit better. All right. All right, so, okay, not able to move this one. All right, so let's just move on to the next structure here. I will eventually move that. And so structure C is the choroid. Okay, so the choroid is a very important layer in terms of how light is moving through or through the eye, right? And so D is what they call the fovea, or otherwise called the yellow spot. Okay, so it's also called the yellow spot. All right. So great. Now we have that. So what we can do here now, right, is look at the question. It said that which part will help to prevent the internal reflection of light? And so the answer here will have to be C, okay? Because the choroid is what is prevent light from being internally reflected. Okay, so option C will be our answer. All right, option C. All right, so now if I could move this a little bit, awesome. All right, so good. Now you see the correct labeling there for aqueous humor. All right, we have different parts of the eye, which, like, for example, the ciliary muscle, the suspensory ligament, the iris, the pupil. You need to know all of that. The vitreous humor right here, the optical nerve right here, and at this point right here, I'm going to put that spot right now. That's the blind spot. And the fovea is where you have the most light-sensitive cells. That where you have the most rods and cones, and you get the sharpest vision at the fovea. We also have the retina, which is this inner part right here. The last structure right here, that's the sclera. All right, so just please remember those structures. So let's go to question 39. And in question 39, it states that what would be the effect of vasodilation and sweating on the body temperatures and on the amount of moisture on the skin surface. Now, what you need to remember, what is vasodilation? Vasodilation is when the blood vessels near the skin becomes wider and they move closer to the surface of the skin to release heat. All right. So as a result of vasodilation, right, the body temperature must get lower, because remember, vasodilation is when the blood vessels get wider, they dilate, open up, and go close to the surface of skin to release heat, right? And while you're releasing heat and you're sweating will also increase the moisture on the skin. So looking for decrease because of vasodilation, and also sweating because sweating also cools the body. So they are looking for a decrease in temperature. So you know that C and D, they will be totally out because they are not increasing the temperatures, right? And the best answer here is B, because the moisture on the skin also increase simply because of the sweating process. All right, let's go to number 40, and we're almost there. Now for question number 40, let me just zoom into this a little bit so you can get a better viewing of the diagram. And so the diagram shows a section through a flower, and so we have the different parts of the flower. This part is called the anther. Okay, remember that's make up part of the male part of the flower, and this piece that connects the anther is called the filament. All right, so that's a filament right there. Okay, and a filament. I'm going to move this a little bit so we could get a better see. That's a filament there. Okay, actually what I want to do is kind of able to label it, all right, with probably a let me use a line to label it. So this piece is a filament here. Okay, and we have three parts here. Right here we have the stigma. Let me try to write that in. Okay, so that is the stigma here. All right, I'm going to type the next one in and go a little bit faster. So that is the stigma. All right, and so from the stigma here we have the next structure here, which is the style. So we have the style, which is this long portion right here. So this is the style. All right, and then we have number 3, which is the ovary, and number 2 here, which will be the sepal, and number 4 here will be our ovule. All right, and so those are some of the structures you need to know. The anther and the filament make up the male part of the flower, which is called the stamen. The stigma, style, and ovary make up the female part of the flower, and that is called the pistil or the carpel. Okay, so that's need to know. All right, so importantly now, it said in diagram shows a cross section to the flower. So what we're going to look at the question now, it said after the flower is fertilized, which part will develop into the fruit and which part will develop into the seed. As you know already, that the fruit is formed from the ovary and the seed is formed from the ovule. So looking for 3 and 4 right here respectively, fruit and flower. Okay, so our option here going to be D. Okay, our option here is D. Let me just highlight that real quick. The ovary forms the fruit. The ovules form seed. All right, so 41, and we're really, really close to the end now. And so 41 state now, the diagrams show pollen grains from 3 different species of plant as they appear under a microscope. It said the diagrams are all drawn to the same scale. So in this case, size matter. Okay, and so look at it carefully. Right, it said which pollen grains are involved in insect pollination. All right, by looking at the diagrams carefully, right? Look at them carefully. What you'd realize, right? What you'd realize is that if it include insect pollination, all right, so if there's an insect pollination, what you need to know is that there must be something to stick the pollen onto the insect, okay? So they cannot be totally smooth. And if you notice option 3, which is probably easiest to identify, notice these wing-like structures, right? So you see the wing-like structures on these. And so the wing-like structures now will allow them to move by wind. So you know that you can't have option 3 within your answer because that's wind-pollinated pollen. So by virtue of this, option C will be out because of number 3, and D is also out. So leave it between option A and option B. But which is our best option here? Option B, which is number 2. Number 2, sorry, I said B, yeah, but number 2, they are really smooth and very small. So to carry it by insect, to be attached to the insect in the first place may be a little bit difficult unless it is stated that I have something sticky. But look at option number 1. If you notice here, you have some spikes on it that can stick to the insect. So here we're going to choose B. So B will be our best choice, simply because the pollen grains will have spikes that they could stick onto the insect itself. Okay. All right. So good. Number 42. It said the diagram shows the female reproductive system, right? And so let me just quickly go through this. And number 3 and number 4, they are ovaries. They are part of the ovaries, of the ovary, of the uterus, right? So 3 and 4, they are part of the uterus. 1 and 2, they are fallopian tube, and number 5 is the vagina. So what this question is asking now, it said, at which labeled point are sperms and ova released? So looking for where the sperm will be released and where the ova will release. All right, and so ova, the ova represent the word for eggs. Okay, so where eggs are released and where sperm cells are released. By virtue of this, if you look at it carefully, right, you'll realize that the vagina is what will receive the sperm cells. So we should know that sperms are deposited in the vagina, which is number 5, and the fallopian tube is where egg cells will be deposited. So looking for number 5 and number 1. And so this will be here, number 5 and 1, which is option C. Okay, that is option C. All right, so let's quickly highlight that one as well. Option C. All right, so let's go to our next question here, which is question number 43. And question number 43, it states that how will the composition of pregnant woman bloods change as it passes through the placenta? So what is happening here to the mother's blood? And it said a pregnant woman as it passes through the placenta. Now remember, the placenta is a filtering organ for blood for the mother and allow substances to go to the fetus. All right, the mother's blood and the fetus and the fetal blood, they do not mix because of a number of reasons. One, because of difference in blood pressure, all right, and also difference in terms of the blood group, because the mother and the fetus could have different blood groups. So if they mix, that 2 different blood groups do not mix. Otherwise, it could cause coagulation or cause malfunctioning of the components within the blood itself, right? Such as high production of white blood cells, and white blood cells start to attack other components within the other blood, and so on. So it's never able to mix. All right. And so 43 here now, we're saying that now the concentration of dissolved nutrients, will it increase or decrease? Remember now, the first thing to remember that the mother is supplying nutrients to the fetus. So therefore, the nutrients within the blood must decrease. So you know that options C and D, they are out. Okay. Now the concentration of urea. Urea is a waste product. So it coming from the fetus will deposit the waste product into the mother's blood via the placenta. So definitely option B will have to be our correct answer, because again, the mother's blood will pick up waste products such as urea from the fetus. The fetus will not urinate nor defecate within the mother's womb. All right, everything goes through the blood. Now let's look at 44 real quick, and we're really close to the end. So 44 here now states that when a baby is born, the following processes occur. One, the breaking of the amniotic sac, and in very layman term, this is when you say the water bag break. Okay, all right. And then 2, we said contraction of the muscles in the uterine wall, and when the muscles are contracting the uterine wall, that's when you say the female is in labor. So the contraction of those muscles will cause labor. And number 3, the cutting of the umbilical cord. That's after the baby is born. Then of course you have to separate the baby from the mother. Okay, so the first thing to happen here is in which order do these processes usually occur? And so the first thing is that the mother will tend to go under labor first. So number 2 will have to be the answer, because that contraction of the muscle wall, the muscles in the uterus. And then the next thing will happen is that the amniotic sac will break. So there has to be number 2, then number 1, and then the last thing will happen is number 3. So there goes our answer here. It is option A for number 44. All right, so now let's go to our next question, which is question number 45. And after this, we only have 5 more questions, and that will be it. Now, we said the graph shows the height of a child from birth to age 10. Okay. And so let's go into the graph a little bit so we can see a little bit better. All right. And so that's the full graph there. So notice you have the height in centimeters and age in terms of years. And so here, now, let's see what the question is asking us. And so question number 45 said, what was the approximate height of the child at age 7? So looking at for the approximate height at age 7. And so let's look at it now carefully. Here, we're going to draw some lines to make it a little bit easier for you. So looking at age 7 and approximate height. So age 7 is exactly between 6 and 8. So we're going to kind of draw a line straight through. Hopefully this line would be straight enough for me. All right, so it will fall right here. Okay, so I'm going straight through. Hopefully this line will be straight. All right, so we know exactly where we're going to end up anyhow. So it's going to be right here, right? So notice this line right here. That's where it is. And then we could go straight across to the other side right here. And let's carry it straight across. You're going to be on this line. So follow this line carefully. My hand is not so straight, but yep, you're going to fall right here, right? Now, what we can do to get accurate values, we can check the amount of spacing between these. So we have 1, 2, 3, 4, 5 lines. And it moved from 100 to 120. So if it's 5 lines, that means each of them will be by 4, right? And so we have 1, 2, 3, 4. And so 4, there's 16. So that seems about 116 there. And so our answer right here for this question, all right, zooming too much. Our answer there will be our B, 116. All right, so this is a simple technique to see if you actually could read graphs properly. All right, so that is done. And let's now go into our next question here. And so question number 46. Now, question number 46, now it states that the diagram shows an experiment to investigate gas exchange in a leaf. Now, what I wanted to observe carefully right here, if you notice, we have substance to absorb carbon dioxide. We have a black cover. We have a leaf. And we have a ink drop. Now, the thing is this. If you cover this with a black cover, what is one of the most likely thing that will happen or will not happen? The leaf is covered with something that is black, right? And so if the leaf is covered with something black, it means that there's no light penetrating. And so the leaf will not be able to photosynthesize. Okay, so in other words, the leaf is in a dark place. Okay, if the leaf is in a dark place, what will more likely happen? Now we said, in which direction does the ink drop, move, and for what reason? So in what direction? So we could either move in or out, right? So let's think about how will this ink drop move? And so will it move to the right? Will it move to the left, right? And so for what reason is it moving for? Now, since the leaf is in a dark place, we know for sure it cannot be because of photosynthesis because there is no light. So we're going to take out photosynthesis. So it must be because of increased respiration. So in what direction will it move? To the right, to the left? Will it move in? Will it move out? And so it will move towards the left, which is towards the leaf, because the leaf will take in the oxygen. Okay, and so it will move towards the what? Left, which is towards the leaf, carrying oxygen for respiration. So our option there will be B. Let's go to our next question. And this is question number 47. And number 47 state that a sample of expired air is collected in a gas jar. Another gas jar contains normal atmospheric air. So we have expired air versus normal atmospheric air. It said a lighted candle is placed inside each gas jar as shown. The time taken for each flame to go out is measured. All right, as the candles burn, they use up oxygen available in the jar, right? And so let's look at both of them carefully. So we have jar X and we have jar Y. So we have the air sample in both. The candle is there. We have a lid. So remember now, as anything that is combusting, anything that is burning will use up oxygen. Now we say the table shows the result of this experiment. So we have gas jar X and Y, right? And so gas jar X and Y is the time taken for candle flame to go out. So X take a much longer time, Y take a much shorter time. All right, so what we can say here now, if X is taking a longer time to go out, what we can say about X is that it contains more oxygen, isn't it? And if Y is taking a shorter time, therefore mean Y contain less oxygen. Because remember, oxygen is needed for combustion, right? And so let's look at this now careful at our options here. And remember, jar X is going out. I want to see all the options. Okay, yeah, good. This is good. All right, so remember, look at the numbers and let's now reason through this. Now we said, look at the explanation of the differences between the result in jar X and Y. All right, and so it's a jar X contain atmospheric air, sorry, which has more carbon dioxide. So if X contain more carbon dioxide, it will take a shorter time, not a longer time. So that is incorrect. If you look at option B, it's a jar X contains expired air, which is a lot of and has more carbon dioxide. Again, that is not correct. All right. Jar Y contains atmospheric air, which has less oxygen, which is not true because if it is atmospheric air, it should have more oxygen in it, right? And so it should take a longer time. So D now is that jar Y contains expired air and has less oxygen. So it is expired air in jar Y and less oxygen because you have less time for burning. And so that looked like the most feasible answer right there. So option D for this question is the correct answer. All right, let's look at number 48, and we have 2 more to go and we'll be finished. All right, so now question number 48. It's a diagram represents chromosome in a cell of a fruit fly. All right, and so if you notice the chromosomes here, they occur in pairs. So you have a pair of chromosome here, a pair right here, a pair right here, and a pair right here. So we have 4 pairs of chromosomes, right? Just going to highlight that. Now, it's how many chromosomes would there be after meiosis? Remember, meiosis produces haploid cells, which is half the number of chromosomes. In other words, 1 of each pair, okay? So it asks how many chromosomes. So in total here, how many chromosomes we have? If we have 4 pairs, that means it's 8 chromosomes, right? And so, therefore, if you have 8 total chromosomes, half of 8 will only be what? 4. So 48 has to be B. Simply because we produce half the number of what? Chromosome. We have 8 or otherwise 4 pairs. So we end up with only 4 after meiosis. Okay? Those are haploid cells. All right, so 49, and we're almost there. Now 49 now is stating that in pigeon peas, the gene which determines the texture of the seed coat has 2 alleles. The allele for smooth seed coat is dominant to the allele for wrinkled seed coat. As a result of cross between heterozygous, notice this key part here, heterozygous plants, 108 of the new plants produce pigeon peas with smooth seed coats. The question asks, how many new plants would be expected to produce pigeon peas with wrinkled seed coats? Now, first and foremost, let's think about this really, really carefully, right? Now, what we need to do is first to understand what is happening here. The fact that they state that both parents or both plants are heterozygous. Remember, if they're heterozygous, it is always a 3-to-1 ratio, which is a 75 to 25% ratio all the time, no matter what, right? So let's look at this now. You can even use your own alleles if you want to kind of draw this so you can actually understand it more carefully. But for the fact that they said that smooth is dominant, right, over wrinkle, what you can use, if you want to do this is unnecessary anyways, because remember, you get a 3-to-1 ratio, but I want to show it if you really do understand it, right? So let's use uppercase S for smooth since it is dominant, and lowercase s for wrinkle. I don't like using S because S tend to look the same. And again, if both parents are heterozygous, let's do a Punnett square real quick. I just want to show you this real fast. So we're going to put a big S and a small s. All right. A big S and a small s. And so you have 2 big S's, a big and a small, a big and a small, and then 2 small s's, right? Hopefully you could see that real carefully. All right, so let me just erase that a little better. So we have here, we have a small s right there, and then here we have 2 small s's. All right, and so what I want you to understand here now is that it's a 3-to-1 ratio. Okay, now what will be in the 75% will be the dominant trait. The dominant here is smooth. Okay, wrinkle is recessive. Okay, and so the question asks how much of the 108 produced will be wrinkle, which is looking for 25% of that. So how you work this out, remember, out of the 4, 3 out of the 4 will be smooth, right? And remember here now we said that 108 will be smooth. So you want to know how much will be wrinkled. Let's look at this. Let's look at this mathematical statement. If 3/4, right, is equal to 108, then what will be 1/4? Because the wrinkle will be 1/4. So I want to know what 1/4 will equal to. If 3/4 is equal to 108, then what is 1/4? Easy way to kind of work this out is simple. Look at it, the numbers, and you're going to apply this concept to it. If 3 out of 4 is equal to 108, what is 1 part equal to? That's what the question is asking you. What is 1 part equal to? 3 out of the 4 is 108. So what 1 part equal to? Simple to get this answer, you're going to divide 108. So let's say 108, right? Divided by 3. Because if 3 parts is equal to 108, that means 1 part must equal to 108 divided by what? 3. And that's how you get the answer. And so 108 divided by 3 will give you what? 36. So 36 will be the answer here. Okay. So let's just highlight that. So A is the answer. All right. Awesome. Again, remember, easy mathematics. 3 parts out of 4 equal to 108. So what 1 part equal to? 108 divided by what? 3. All right. So I hope you get that, understand that. Now let's go to our last question. And our final question here now, nice little table. Let's get that all together in one Zoom. So we get everything one time here. All right, that could work. All right, so here now is asking us now. It said that the table, matter of fact, shows the amount of water and salt lost from the kidneys and skin on a hot day and on a cold day. It said food and drink intake are the same on both days. So you don't do anything different, right? So since nothing was done differently, now it asks you, now what do these results mean? Now one observe these results carefully. First and foremost, water loss from a kidney. And on a cold day, you have more water loss from a kidney. Less water loss from the kidney on a cold day, right? And there's on a hot day versus cold day. And there's a reason for that. On cold days, you sweat less, so you urinate more. On hot days, you sweat more, so you urinate less. Now, water loss from the skin. On cold days, you do no sweating. On hot days, you sweat, so you have more water loss by sweat or loss from the skin on hot days versus cold days. Now, salt loss from the kidneys. Cold days, we have 20.2, all right? And salt loss from the skin on cold days is 0 because remember you're not sweating, so there's no salt lost. But all the salt will be lost through urination. Hot days, the salt lost from the kidneys is 14.4 that is shown right here. The salt lost from the skin is only 5.8. So what are these results showing? What can we get from this? All right. Let's look into it and see. All right, let's see option A. Said less water is lost from a kidney on a cold day than on a hot day. This answer cannot be correct because you notice the first column, water loss from a kidney on a cold day is more than on a hot day. So that's not true. Okay. All right, so and this is saying less water is lost from the kidneys, which is not true. More water is lost from the kidneys on the cold day. Now let's look at option B. Now option B states that more salt is lost from a kidney on a hot day than in a cold day. Now let's look at the salt that is lost from a kidney. Now the salt that is lost from a kidney, let's just kind of highlight them real quick. I'm going to put that one there. And so the total salt loss on a cold day, it is 20.2 plus 0, which is 20.2. And now let's look at the salt that is lost on a hot day. Let me just put it in a different color so it'd be easier for us to identify what you're talking about here. All right. And so, yep. So on this day, if you notice it, if you should add 14.4 and 5.8, you still get 20.2. Okay. So that's now interesting. So for option B, that state that more salt is lost from a kidney on a hot day than on a cold day is not correct because both are losing the same amount of salt. Going across on a cold day, it is 20.2. Going across on a hot day, the same amount of salt was lost. A combination from the salt lost from the kidneys with the skin. So option C is saying that the total amount of salt lost each day is the same, which is truth based on the amount that we see here. Let's look at option D. Water is not lost from a kidney on a hot day, which is not true, because on a hot day, you have some amount of water that is lost, which is 0.4. So option C is our correct answer. And this is our final question, and so on. I want to tell you that I am so happy for you to be here. I know we spend a lot of time going through these questions, but study well, work hard, and do good on the exams. Take care until we do our next review.